Today's talk is entitled Advancing Cancer Research from Basic Biology to Organoid Innovations. And this is presented by Professor Mandeep Kaur, who is a senior academic within the School of Molecular and Cell Biology. And Mandeep is marrying the clinical aspects, so clinical samples, with basic science, with high-end, high-tech basic science approaches, using these miniature organs, if you like, to model colorectal cancer processes. We're going to follow with a series of 3 talks in total, the first one by Mandeep, and then the next 2 in succession given by her postgraduate students. I think I'll leave Mandeep just to introduce her students once she's done her presentation. And then we'll take questions at the end of the series of the talks. So hold on to your questions until the end. So over to you, Mandeep.
So I'm really honored to be here today, and particularly more honored because I'm going to share this stage with 2 of my very talented PhD students today. So this is going to be a team seminar, and we are going to showcase the work that we are doing at Wits for past 6 to 7 years. So before I move on to actual presentation, I would like to talk a little bit about the work we do, the overall scope of our work. We use a lot of mainly experimental wet lab work, which is supplemented by bioinformatics work in our lab. And we are very focused on understanding cancer molecular mechanisms and developing new anti-cancer molecules. We do a lot of work into understanding drug resistance. That is one of the themes for our research. And we develop biomarkers, both based on bioinformatics work as well as we do experimental validation. And we also look at metastatic processes in cancer cells. So today we are going to present a part of our overall work. Of course, not everything, there is no time, but just a part of what we do. So we all know that cancer is an increasingly, it's an increasing burden all over the world. In 2022, there were 20 million new cases they were identified, and there were about 10 million deaths that happened due to cancer. And the damage is not only in terms of the incidence or mortality rate, but the economic burden of cancer is significantly higher as compared to many other diseases. If we look at just at South Africa, this is an old number about 10 years ago because I couldn't find a new number here, but the highest cost per cancer death was noticed for South Africa. That was $2 billion out of all BRICS countries, which is quite huge considering our population size and our resources. And according to one estimate, by 2050, the global economic cost of cancers would rise to $25 trillion. And the complicating factors are not just the prevalence and the mortality, but also the drugs that are available in the market, access to different types of drugs, and also, particularly in Africa, we have a problem in terms of late diagnosis, which actually puts our patients at a very high risk because by the time the cancer is detected, there is no treatment available for that advanced set of cancers. And if the drugs are very expensive, being in Africa, our patients cannot afford them. So this is one estimate that shows that UN has lately declared that antimicrobial resistance is going to be next emergency for us to handle, for scientists to handle. And in this case, you can see the current death rate is 1.3 million, and by 2050, it is expected to rise to 10 million. But if we look at cancer, we are already there. And you can imagine that what would happen by 2050 if the cancer is increasing at the same rate, and a 77% increase is already expected. And approximately 70% of deaths from cancer, they occur in low to middle income countries, such as in African countries. And to make situation worse, about 90% cancer-related deaths are contributed to drug resistance if patients have access to medications. So there are multiple problems that we need to tackle if we really want to find cure for cancer and we want to get rid of this disease. Oh, sorry. It is just slow, please bear with me. I was expecting too much of this. Okay. So if we look at drug discovery and development pipeline, I don't need to talk about it a lot because we understand that even any drug that we want to develop and bring to market, it takes about 12 to 15 years. And the most important aspect of drug discovery and development pipeline is the preclinical step, where we validate the molecules that we have identified, we characterize them, we do a sort of preclinical testing using computational methods, using in vitro methods, using in vivo models, and so that we are sure that we are okay to take these medications or these new molecules specifically to phase 1 trials. And there are lots of rules are in place for this. For example, like for preclinical step, if we want to take any molecule to phase 1, we have to do at least 3 animal model studies. So that is the requirement by FDA or the regulatory agencies. But if we look at different models that we have available and the drug discovery efforts that are being done using existing models. So this graph shows you the likelihood of approval from phase 1 of clinical trials. And if you look at oncology, that is has the lowest likelihood of approval based on the existing methods that we have. And that is just 5%. And this is something like $1.75 billion drug development that we are talking of. And after preclinical testing, if we have only 5% success rate in phase 1, which is severely limited. So it means we need to relook at exactly what we are doing. What type of models are we using at the preclinical stage so that we improve on our chances in terms of getting better results when molecules reach phase 1. So if we look at evolution of different disease models, then you can see that 2D monolayer culture, the normal cancer cell lines, standard cancer cell lines that I think everybody is using in our laboratories, they were actually established in early 1900s, but they came into mainstream use in 1950s. And we still have very heavy reliance on these models. And then by 70s, so little bit more physiological. I think this is, something is wrong with this one. Let me just move on the other side. Simply doesn't want to move. Yeah. No, I'm pressing the right one, but it's not moving. Yeah, okay, let me try. Sorry about this. So 2D monolayer cultures on a membrane, they were established in 1970s. 3D spheroid cultures, again in 1970s, but in past 2 to 3 decades, there has been a lot of use that is coming up for spheroid cultures. Animal models are there even from 6 BC. So people are using animal models for a very long time now. And lately, since 2009, 3D organoid culture models are coming up in the mainstream because first landmark paper was published in 2009. So these 3D organoid cultures are known as mini organ cultures, and they recapitulate the biology of a particular organ from which they are grown. So this is what we are going to focus on in the later half of this presentation. And if we look at other preclinical disease models and their suitability in terms of preclinical testing, then you can clearly see that 2D cell cultures are very suitable for high throughput screening. They are simple, easy to use, but certainly they do not capture the physiology of basically human physiology if we talk here. Similarly, there are issues with non-human primates and and mouse. But out of all these factors, human organoids have been found to be so far the best models that are available out there. So, in a sense, cancer research relies on using various types of models that are available to us to understand disease biology for therapeutic applications. And the question we asked in our lab, that while using all the available different types of preclinical models that we have in existence, can we work around a concept that can cholesterol lowering be used as a universal anti-cancer approach? So my lab heavily focuses on understanding the role of cholesterol in cancer, how it fuels cancer, how it imparts drug resistance to cancer, and can we use cholesterol depleters or cholesterol lowering agents as anti-cancer therapeutics? So I would just briefly like to touch upon different mechanisms of cancer drug resistance that you can see here. A lot of them are listed here, and my lab is focusing on these 5 of these, which are with the green tick. And cholesterol homeostasis is picking up. This concept was not in the mainstream about 10-15 years ago, but lately in past 5 to 7 years, a lot of work has been put in by very large laboratories across the world, because more and more proofs are coming in that cholesterol imparts severe drug resistance to cancer cells. So cholesterol's role in health, it's very important. It's not all ugly as has been told to us, but if cholesterol is not there, we basically cannot survive. This is the second most abundant molecule in our body after glucose. So you can imagine that how important it is. It is responsible for vitamin D production, cell membrane formation, hormone production, especially male and female hormones, bile production, nerve cell insulation, immune cell function. So it performs a lot of different functions in the system. And that is why our bodies don't rely on outside cholesterol. Body likes to produce its own cholesterol, and we only take about 20% from outside. And 25% of body's cholesterol is found in brain, because we know that brain is very dynamic organ. It has to be functional all the time. So it has very heavy reliance on cholesterol as well. So just to explain a little bit on cholesterol homeostasis so that you get more comfortable with different gene names and symbols that we are going to talk about going forward. So in terms of cholesterol homeostasis intracellularly, there are 4 processes mainly involved in the process. First, of course, synthesis. When the cholesterol levels are down in the cell, automatically SRABP proteins, they sense the level of cholesterol and they activate and they actually push synthesis of cholesterol or activate synthesis of cholesterol in the cell so that optimal levels are maintained. The second process is influx. Cells also take up cholesterol from outside if there is the level of cholesterol is low inside, and that is mediated by LDLR receptors. And part of this is recycled to membrane, and then we have free cholesterol inside the cell. This free cholesterol is then stored inside the cell for later use. And this is a very important part where cell stores this cholesterol in the form of cholesterol esters, because free cholesterol can be toxic to the cell itself. So a protein named cholesterol ester transfer protein, it plays dual role in our body. It helps to store cholesterol inside the cell, and it also helps outside the cell in bloodstream to clear cholesterol from the system. And the fourth aspect is where cholesterol is effluxed out of the cell. When the optimal concentration has been achieved, homeostasis has been achieved, it will just push out extra cholesterol or efflux extra cholesterol so that inside the concentration of cholesterol is maintained and cell can go on. So deregulated cholesterol in cancer has been linked to multi-drug resistance, because what has been found that not all cancer, but some types of cancers including breast, prostate, esophagus, colorectal, pancreatic, these cancer cells have been found to be about 6 to 8 times richer in cholesterol. So they keep on accumulating cholesterol so that they can divide faster, cancer can progress, cancer can migrate, and cancer can be more invasive. And how do these cancer cells do that? They increase the cholesterol biosynthesis and uptake, they decrease the cholesterol efflux, they increase lipid droplets, the stored form of cholesterol, they increase the number of lipid rafts. So lipid rafts are these small domains, 10 to 200 nanometer domains in the plasma membranes, and they act as platforms for various signaling processes. And they are housed in the cholesterol. So, and they are very protected by cholesterol being there. And all these increase the membrane rigidity and reduce permeability, which leads to multi-drug resistance. So this is the concept around which we are working, and we are trying to find ways to lower this cholesterol level so that we can crumble cancer cells and they must not simply proliferate. So when we talk about cholesterol lowering agents, the first thing that comes to our mind is statins. I think all of you must be aware of statins. These are blockbuster drugs. Every 1 in 4 person takes, globally, takes statin. So they are so famous and right now. But the problem comes, if we have to use statins, then there is a concept called as statin intolerance. Means there are people who do not respond well to statins due to multiple reasons. And it is overall prevalence of statin intolerance was found to be something like, different studies said 15 to 20%, but one very large meta-analysis on more than 4.2 million people came out. And what they found out, this is very interesting, that if people who, like black race, people do not, there are 30% people who do not respond to statins at all. They show statin intolerance. And in women, in females, this percentage is 48%. So imagine if statins are coming as breast cancer treatment, about 50% of females won't even respond to statins. So that is the reason that there is a need to look for new cholesterol lowering agents. And that is where we are working to develop a new class of compounds. This is not a new class, but we are giving it a new role as anti-cancer drugs. So these are cyclodextrins. So cyclodextrins is a class of compounds. They are used as excipients for about 35 marketed drugs already. They, even in your eye drops, your skin creams, even in your food, you take cyclodextrins every day. So cyclodextrins, they have been found to be extremely safe for human consumption. They are used as excipients, means they solubilize different drugs and make them bioavailable for their actual functions. But what we are trying to do is we are trying to use a particular cyclodextrin, which is 2-hydroxypropyl beta cyclodextrin, which I will refer to as HP beta CD, as new anti-cancer drug. And we have done a lot of work around this molecule that I'm going to show. So how this class of molecules work, that they have a structure like a cavity. The inside is hydrophobic, outside is hydrophilic. And that is how they can capture all lipids in them, hormones. The cavity is of this size that they can capture hormones as well. They can capture cholesterol. So they bind to cholesterol and remove cholesterol from the cellular membranes. And HP beta CD can also enter the cells, and it removes cholesterol from within the cell. And it also removes lipid droplets from within the cell. So this is what we are exploiting for our work. So now I'm going to talk about some research that we have done, but before that, just to again make you familiar so that you don't get confused with different types of cholesterols that I'm going to talk about. We measure free cholesterol using a sort of blue dye, which is Philippine. Cholesterol esters means the stored form of cholesterol using, you will see, using body fluorophore, which stains it green. And then we have Alexa fluor, which is red in color. So it actually labels the lipid rafts, and then we can measure the level of cholesterol in the membrane. So this is for the membrane, this is for the stored form, and this is the free cholesterol which is present inside the cell. Okay, so the first investigation that we started was in breast cancer. So this work I started sometimes in 2016. We first did basic cell line work where we used MDAMB231 and MCF7 cells, which are triple negative breast cancer and estrogen and progesterone responsive cells, respectively. So what we found that when we treated these cell lines with HP beta CD, we saw decrease in level of cholesterol of all different types. We saw an increase in the apoptotic death of these cells, encouraging initial results, but main encouragement came from the very first animal study that we did. What we did, we divided our animals into 3 different groups, depending on the size of the tumor, and then we saw that at the very late stage, with 10 doses of HP beta CD, we found that there was a tumor reduction of 74%. In the intermediate stage, the reduction was 94%. And in the very initial stages, when the tumor was just 20 millimeter cube, 20 to 30, just 10 doses of HP beta CD actually completely eliminated tumors. And then we kept those animals on relapse, and we could not find, again, like there was no relapse, so there was no tumor coming back. So then we did some further studies on this to look at drug protein interactions of this HP beta CD, and these proteins were selected, or these genes were selected based on the arrays, RT-PCR arrays that we run. And then we selected some of these proteins to test for drug protein interactions. And out of that, we found that SFRP1 was the main protein that we found that this molecule was binding to. And SFRP1 is a natural negative regulator of Wnt pathway, which is a very important and well-investigated pathway, which is responsible for many different types of cancers, specifically colorectal cancer. Then we also looked at CETP, because I told you CETP plays a role intracellularly as well as extracellularly. So because it helps to store cholesterol, so we tested the hypothesis that if we just remove or knock down this particular gene from cancer cells, how cancer would grow. So we knocked down CETP, we injected these cells in mice, and here you can see that without any drug treatment, automatically there was a tumor reduction, or tumor didn't grow in 87% of cases, or there was a reduction of 87% without any medication. So that confirmed for us that yes, cholesterol is playing a big role in breast cancer. As well as several markers in the cholesterol pathway can actually help us understand it, and they can act as markers of cell survival or cancer survival. Then we are also working on another ingrown or in-house grown cholesterol depleter and acetyl plumpagin. When we combined that with tamoxifen in CETP, after CETP knockdown, then you can see the level of apoptosis is markedly increased. So the cholesterol depleters can work synergistically with existing chemotherapeutic drugs to enhance their effect as well. And in terms of different pathways, we saw that drug resistance was reduced, cholesterol efflux pumps were increased, lipid rafts were decreased. So all the processes were basically reversed that were fueling cancer cell growth. And then you can see here that several genes involved in drug resistance pathway were also downregulated. So of course, our confidence went up. The next study we didn't want to just focus on tumor reduction or tumor inhibition. We wanted to see if this effect of HP beta CD continues during EMT state, which leads to metastasis. So EMT is epithelial to mesenchymal transition, which is the first step when cells they get ready to spread across the body for metastasis. And in this case, you can see that we did this in one of a very well-known mouse memory model, NMUMG and MCF7 cells. We saw a reduction in the expression of marker vimentin, which is a marker for mesenchymal transition, for the mesenchymal state. So it means that this molecule has the ability to block EMT in cancer cells. And then we also looked at several different transcription factors which facilitate epithelial to mesenchymal transition. All of them, they were downregulated. Then we also looked at the invasion of the cells. It was significantly reduced. And we also looked at the drug resistance, like PGP proteins, which are the drug, basically drug transporters. So we saw that their function was also decreased. So height of these bars is actually inversely proportional to the PGP activity. This is how this assay works. So here you can see here that in the post-EMT you can see that PGPs are active, but after HP beta CD treatment we saw that their function was blocked. So it means that HP beta CD is capable of reducing drug resistance. So after getting these results, then we wanted to see if it is just this effect is visible only in breast cancer, or can we actually translate everything into any other type of cancer. And that is where then we started looking at colorectal cancer. But then what we did in this case, in colorectal cancer we combined HP beta CD with commonly used therapeutic drugs, 5-fluorouracil and oxaliplatin. So oxaliplatin is very toxic drug, and mostly it is given to even breast cancer patients when they are at metastatic stage. But what we observed that the dosage of oxaliplatin, when we gave it to animals, animals could only take 6 doses of oxaliplatin and they all died. But when we combined oxaliplatin with HP beta CD, we could give 12 doses of oxaliplatin along with HP beta CD. So that shows that HP beta CD actually reduces a lot of side effects linked to these chemotherapeutic drugs. And then we also saw that the liver markers, in this case, they were still normal. There was no metastasis in animals. When we looked at different markers or different genes responsible for cholesterol metabolism, so blue shows that they are sort of downregulated. In terms of Wnt pathway, as I mentioned, it's very important for colorectal. Again, we saw a significant increase in SFRP1, and we also saw a decrease in several genes which are linked to Wnt pathway. So this is just a depiction of Wnt pathway. SFRP binds to Wnt, and then it blocks the whole process that happens here that leads to beta-catenin accumulation in the cell. But when SFRP binds, then beta-catenin is degraded and cancer cannot proliferate. So this tells us this is the mechanism how this molecule is actually working. And then we saw in terms of cell death that the combination of oxaliplatin with HP beta CD in our cellular models also increased apoptosis, and then we also saw significant reduction in the drug resistance as we have seen with the breast cancer. Again, in terms of, I won't spend much of time here because I spoke about it. So similar results that we got for breast cancer and EMT, we could actually reproduce the same results in our colorectal cell lines. And here you can see, again, there was an increase in PGP, reduction in PGP activity when HP beta CD was combined with Folfox. Apoptosis was increased and invasion of cancer cells was also decreased. So till now we were extremely happy and we were jumping around. We thought, okay, we got a very good new molecule, right? This is how researchers work. They get quite excited. So just to put it together, both in breast and colorectal, cholesterol was lowered, tumor was reduced, no metastasis was seen, EMT was reversed, drug resistance reduced, and less invasion. What more do we want? So because we were super confident at this stage, we decided, okay, why not to try it on something else? Let's try pancreatic cancer. And that is where we were just brought back to earth. Because pancreatic cancers, they are highly, highly resistant. So when we started these studies, in vitro, yes, we could see some sort of results with HP beta CD, but those are cellular models. But when we took this HP beta CD treatment in an in vivo model, like this was done by Tasvi and helped by Liz Marie here, and then you can see here with HP beta CD, we could, out of 4 animals, in 2 we could already see metastasis, which we had not seen in previous studies. And even in the combinations, we could see there is metastasis happening. So we were not sure what is happening here. Is the dosage not working, or what is happening here? We are still trying to figure it out. This is the latest work that we have done. We haven't even published it. This is just Tasvi has wrapped up her thesis at this moment. And then you can see here that tumor reduction was also moderate when HP beta CD was combined with 5-fluorouracil, while in colorectal cancer it went up to 75%. In terms of tumor volumes, you can see here nothing much is changing between untreated and HP beta CD. If you look at the gene expression in the tissues, then this is HPCD section at the end. You can see cholesterol uptake and synthesis is increased. And then we can see, like, other genes here, they are downregulated. So it means at molecular level, some changes are coming after HP beta CD induction, but they are not being translated phenotypically when we actually see tumor growth, when we see EMT. So it is trying to work, but it is unable to give us the same effect that we were able to see in breast and colorectal. And similarly, we could actually confirm that in the protein, using protein as well. Okay. So this slide shows you the level of different types of cholesterols that I mentioned earlier in the tissue samples of the tumor that we extracted from the animals. And if you look at this graph here, this is HP beta CD, this is a lipid rafts, and this is untreated. You can see a very significant increase in the number of lipid rafts. So the plasma membrane cholesterol is actually increasing when we treat animals or cells with a cholesterol depleter. Although this is what we were not expecting. Similarly, not much difference was observed in the stored cholesterol or in the free cholesterol. So then we wanted to see if this is the same thing that we are going to see again in EMT, what is happening. If HP beta CD is not able to reduce or the tumor size, maybe it is able to prevent metastasis of cancer. Here you can see mesenchymal marker, vimentin, which is significantly upregulated in post-EMT stage. And you can also see their cell count has increased significantly. When we look at the gene expression profile, then again you can see here, these are the EMT markers. All of them, they are upregulated, although we were expecting the opposite. And you can also see that all different types of cholesterol influx, efflux, and even the synthesis, SREBP proteins, they are highly upregulated. So it means that after treatment with HP beta CD in pancreatic cells, we actually saw an increase in cholesterol synthesis, cholesterol storage. We also saw like increase in lipid rafts, which is basically the plasma membrane cholesterol. So, which means, and also we saw increase in markers of mesenchymal transition. So all that tells us that when we treat pancreatic cells with cholesterol depleters, and this is not just with HP beta CD, that is also true for statins, that their metastatic capability is increased and they become more aggressive. So now this is completely opposite to what I have shown you before, right? So what we learned from these, like using all these models, you can see we are getting different results. And this is not only dependent on just on the models that we are using, it is dependent on the stage of the disease that we are capturing. And we will wrap it up towards the end that why we are seeing these results. So highly resistant cancers, they have high cholesterol needs and they trigger cholesterol synthesis, storage, influx pathways just to stay alive. Their complex biological underpinnings are completely unknown, and these drugs, cholesterol-lowering drugs in pancreatic cancer patients with advanced disease need to be avoided. And this is not a conclusion just from our work. This is a conclusion, there are studies, there's one study published from Fox Chase Center in US, and they have also shown, actually the person who is, he's a clinician and he has said that he's already watching very carefully if his patients are already on statins or not. So they must not continue on statins, especially patients who have pancreatic cancer. And we are going to show you that we are, in our organoid models, we are seeing the similar thing in advanced, or you can say more aggressive colorectal cancers as well. So there are certainly limitations of this cholesterol lowering as anti-cancer strategy. As I mentioned, cancer type and stage, different types of mutations that are present in these cancers, they also impact how cells react to or tumors react to these cholesterol depleting strategies, and of course type of model under study. So based on this, what's next for us? What we want to do next? Where we want to take our work? So we definitely want to understand that what triggers high dependence of some of these cancers to cholesterol lowering agents, because their clinical implications are huge. Specifically, statins are so widely used in the populations. And then we want to evaluate chemotherapeutic response or non-response in South African intestinal organoids. So these are the latest models that we have established in our laboratory. And next year we are going to do single cell sequencing from these samples along with organoids. And we want to understand what is going on in South African patients. And of course, like these models, they do bring enhanced research capability for us. Just because, you know, they recapitulate the biology very well. They are in 3D format. We can study them physiologically as well. They give real-time results, and on top of it, they will help us to eliminate animal models going forward. And of course, realizing the field of precision medicine. So this is where I'm going to stop. I'll come back again, but now I want to invite Ruth. Ruth is a senior PhD student in my lab, and Ruth and Naaziyah, both of them, they are working together. We are working on this concept for something like 2 years now. We have had our lots and lots of hurdles, but now we are at a stage where we are very comfortable in growing organoid models, and Ruth and Naaziyah have done a lot of work in the lab around that. And they are going to tell you all about organoids and some of our baseline results that we have generated so far. Ruth, over to you.
My name is Ruth Aronson. I'm a PhD student of Profs, and I'll be taking you through the initial data that we have generated with our 3D organoids. So Prof has introduced organoids, but just on a molecular level, what are organoids? Organoids are a 3D cell culture system derived from stem cells from any organ that can then recapitulate both the structure and the function of the organ. The organoids that we are currently culturing are intestinal organoids, taking a closer look at the microscopic structure of the intestine. The intestine is made up of a crypt-villi axis. At the base of the crypt, this is rich in intestinal stem cells, and this is key for the growth of our organoids. These stem cells differentiate along the villi into all of the cell types present within the intestine. Cell types such as enterocytes, which are responsible for nutrient absorption, goblet cells, which are responsible for mucus production. We have Panner cells, also at the base of the crypt, which will support our stem cells, and other cells such as enteroendocrine cells, which is found in a really small population, but very important for immune response and hormone production. Now, these organoids can recapitulate this crypt-villi axis and form this heterogeneous structure expressing all of the cell types present within the intestine. And also something that's quite amazing about this model is that it can be used for co-culturing with either immune cells or with microbes, so we can take a closer look into the immune response or infectious diseases. So this is a nice workflow showing all of the milestones that we have achieved over the last 2 years of culturing our organoids. First, informed patient consent is obtained, and the tissue is then brought back to our lab where it is digested, crypts are isolated, and then we can develop these organoids. Once we have organoids nicely established and developed, at this point in time we can culture them almost comparably to cells, where they need to be passaged, upscaled, stocked, and then also thawed, and then from that point on they are ready for experimental use. So this takes a closer look into the workflow or how we generate or establish these organoids from patient tissue. So starting off with washing the tissue back in our lab, the tissue undergoes a series of both mechanical and enzymatic digestion to the point where we can isolate and visualize the crypts. And just to refresh, that the crypts house the stem cells that are necessary for the growth and establishment of our organoids. The crypts are then filtered through. Any presence of red blood cell is removed and then resuspended in a basement membrane extract. This has key ECM extracellular matrix proteins that are necessary for cell-cell ECM interactions, as well as the basement membrane extract will allow for the 3D structures to form. Then media is added, which has all of the necessary growth factors needed to allow for the expansion of stem cells. So this here is a slide showing organoid growth dynamics in a matched patient sample of normal versus cancerous organoids. As you can see in this particular line, the organoids grow really well and proliferate greatly over a period of 8 to 10 days. As you can see in this particular line, the cancerous organoids are growing a little bit smaller than the normal organoids, and that is completely patient dependent or dependent on where we get the sample from, the tissue from. Okay, so the next step is, once we have these organoids established and growing, we need to characterize these organoids before we jump ahead to any downstream analysis. And we need to do this to ensure that the tissue, the organoids, is of intestinal origin and that it matches the patient tissue. So the first analysis we did was H&E staining, hemotoxylin and eosin staining, which gives us insights into the microanatomy of the tissue and the organoids. And as you can see, particularly within our normal organoids, we have really nice differentiation of our organoids showing presence of lumens, crypts, epithelial cells to match that of our normals. And as I said previously, our cancer organoids are a lot smaller, but we still see this presence of epithelial cells compared to matching our cancerous tissue. Okay, so the next thing we went ahead to do was gene expression analysis using key markers that represent intestinal cell types. So we've got markers like EpCAM, lysozyme, vilin. These are all markers that show us the presence of intestinal cell types. And as you can see, within our organoids, we're having really good expression of these markers in comparison to our tissue. And this was validated in both normal organoids and just briefly our cancer organoids, as you can see. Okay, then our next form of characterization was protein expression analysis. We performed immunohistochemistry staining, which allows us to visualize the presence of these proteins. The first marker we looked at was EpCAM, which is an epithelial marker. As you can see by green fluorescence, that our tissue is of epithelial origin, and this was validated, and our organoids were validated to express EpCAM to be of epithelial origin as well. The next marker we looked at was vilin-1. Vilin-1 is a marker for enterocyte cells. Enterocytes are present in approximately 80% of the population. And you can see that there's really high expression of vilin-1 in both our normal and cancerous organoids. And something which is pretty cool for us to see is when literature comes to life, the fact that enterocytes shouldn't be present at the base of the crypt. And here you can see that is represented nicely within our tissue, that the expression of vilin-1 only starts at the villi, not at the crypt. And you can see high expression also here along the villi of the organoids. The next marker we looked at was MAC2, which is a marker for goblet cells. Goblet cells are present in approximately 20% of the intestinal population, and so you are seeing a lower expression of MAC2 in comparison to villain-1, but that is to be expected, and we are definitely seeing presence and differentiation of goblet cells. And the final protein that we looked at was chromogranin A, which is a marker for enteroendocrine cells. These cells are present in less than 1% of the population, and so we were very excited to see that we managed to get differentiation of this small population, the cell population within our organoids. So we've got one cell here within our normal organoids, and we've got two cells here within our cancerous organoids. So overall, this concludes the characterization of our organoids, and I'll invite my colleague Naaziyah to come up to present the data that we have generated on cholesterol profiling of these organoids.
My name is Naaziyah, and I'm a PhD candidate in Prof. Kaur's lab. As Ruth explained, we successfully characterized these organoids, showing that they were rectal in origin, and the next step for us was to profile the cholesterol levels within these organoids in order to understand where they're at. So we subjected the organoids to cholesterol modulation experiments, where we either supplemented them with 50 micromolar cholesterol for 24 hours, or we treated them with 10 millimolar HB-beta-CD or 1 millimolar M-beta-CD for 24 hours. What was really interesting to note was that in our normal healthy organoids, we saw that treatment with cholesterol and HB-beta-CD did not drastically affect the free cholesterol levels. However, treatment with M-beta-CD did lead to a significant decrease in free cholesterol. When we considered the cancerous organoids, this was quite surprising for us, but we saw that all treatment conditions increased free cholesterol levels, with HB-beta-CD inducing the most drastic increase in free cholesterol within these cancer organoids. Moving on to the lipid droplet cholesterol profile, a similar trend was observed, again with M-beta-CD seen to lower the lipid droplet cholesterol level within the healthy organoids, whereas when considering the cancerous organoids, we see an increase in lipid droplet profile with HB-beta-CD again drastically increasing lipid droplets within these cancerous organoids. We lastly looked at the lipid draft cholesterol levels, and this is where things got interesting. So we saw that treatment with both HB-beta-CD as well as M-beta-CD, it significantly lowered lipid draft cholesterol levels, which does make sense because these cyclodextrins are seen to act at a membrane level. But again, what was shocking for us to see is that in the cancerous organoids, we're seeing this drastic increase in lipid draft cholesterol profile when treated with either HB-beta-CD or M-beta-CD. So in order to understand this data at a mechanistic level, we completed some RT-qPCR experiments. And we looked at genes that are involved in, sorry, it's just being slow. We looked at genes that are involved in cholesterol synthesis. So we looked at SRABF1 and SRABF2, which are transcription factors, as Prof explained previously, HMGCR and HMGCS that are involved in the mevalonate pathway. In order to assess uptake, we looked at LDLR, which will take up cholesterol, but PCSK9, which serves as a negative regulator of LDLR. Genes involved in storage include ACAT, LCAT, and CETP, which directly correspond to our lipid droplets. And for efflux, we assessed APCA1 as well as ABCG1 expression. So just as a reminder, the table on your left indicates the cholesterol profiles that we observed post-staining. And we see a decrease in free cholesterol, an increase in lipid droplet, as well as lipid draft cholesterol following supplementation with cholesterol. And the decrease in free cholesterol, as Prof previously mentioned, could possibly be because excess-free cholesterol is quite toxic to cells. So in a healthy state, the cell will choose to get rid of this excess-free cholesterol. And this is well supported by the increase in APCA1 expression that we're observing within our organoid models. The increased lipid droplet profile is also well supported by the increase in ACAT and CETP over there in the healthy organoids. However, when we look at the cancerous organoids, we are seeing an increase in LDL. So the increase in LDL is well supported because we are supplementing these cancerous organoids with cholesterol. So we would be expecting them to take up the cholesterol. What was really interesting for us to observe is that in addition to a cholesterol uptake, we also saw an increase in cholesterol biosynthesis genes, such as HMGCR and HMGCS, pointing to the heavy reliance of this cancer type on cholesterol for its function. Importantly, despite observing both increases in uptake as well as biosynthesis, we also observed an increase in cholesterol efflux pumps, APCA1 and ABCG1, again pointing to the toxic nature of cholesterol accumulation within the cell, which could serve to be counterproductive for cancer cell proliferation as well as survival. Moving on then to HB-beta-CD treatment. So again, the table on the left is just a reminder on the profiles. We observed increased free cholesterol, a decrease in lipid droplet, as well as a decrease in lipid draft cholesterol. In the healthy organoids, we see that the increase in free cholesterol is possibly attributed to the increase in cholesterol biosynthesis, as we are seeing high levels of HMGCR as well as HMGCS in these groups. But again, because this is a healthy state, we are seeing that compensatory increase in efflux pumps such as ABCA1 as well as ABCG1. Which could be counteracting the accumulation of free cholesterol within the healthy organoids. When we look at the cancerous organoids, we're seeing drastic increases across all profiles. And again, pointing to cholesterol biosynthesis, we see upregulation of genes such as HMGCR as well as HMGCS. What's very noteworthy to point out here is despite seeing these drastic increases in cholesterol biosynthesis genes, our cholesterol efflux genes, ABCA1 and ABCG1, are actually present within near normal levels to untreated cancerous organoids, hence supporting that increased cholesterol profile that we are observing within our HB-beta-CD treated organoids. Lastly, looking at M-beta-CD, so treatment with M-beta-CD was seen to lower free cholesterol lipid droplet as well as lipid draft cholesterol profiles in the healthy organoids, and we posit that this is possibly due to our cholesterol efflux, as we are seeing very high levels of the efflux pumps within the healthy organoids. Whereas when we look at the cancerous organoids, we see increases across cholesterol profile, and this could possibly be attributed to an increase in cholesterol uptake and a more heavy reliance on cholesterol uptake as opposed to cholesterol biosynthesis within this treatment modality. Again, what's really important to point out is that our ABCA1 and ABCG1 present to be within near normal levels of our untreated cancerous organoids, hence supporting that increased cholesterol profile. So in summary, when we subject these cancerous organoids to cholesterol supplementation, we see that the cancerous organoids, in addition to taking up free cholesterol from the cellular environment, they also display an inherent reliance on cholesterol biosynthesis to support the increased cholesterol profile. When we look at our 10 millimolar HB-beta-CD treatment group, this treatment group is seen to be more heavily reliant on cholesterol biosynthesis. And lastly, considering our 1 millimolar M-beta-CD group, as opposed to HB-beta-CD, the 1 millimolar M-beta-CD group is seen to trigger a more strong response in cholesterol uptake. So it's very important for me to point out that all of the results that I have presented thus far was taken at a single time point, and that is 24 hours post-treatment. However, we do know, based off of this discussion and also many published papers, that cholesterol metabolism is quite dynamic in nature. It is a complex and adaptive process. And so the next step for us now is to try to understand whether the result of our treatments is due to the treatment effect or if it is a compensatory response. And in order to achieve this, we are now looking into lifestyle imaging of our organoids in order to understand the treatments and the effects of the treatments in greater detail. So in addition to this organoid work, we chose to cross-compare the results obtained within our work to work conducted previously on 2D models as well as in vivo studies. And for the cross-comparison, we used HT29 cells, which are well-represented colorectal adenocarcinoma immortalized cell line. And we can see that the converse of what was observed in the organoids is presented in the HT29 cells. So we see that when we treated the HT29 cells with HB-beta-CD, we observed a decrease in free cholesterol, a decrease in lipid droplet, as well as a decrease in lipid draft cholesterol. And when we subcutaneously injected these cells into mice, we saw that there was a non-significant decrease in tumor size. We also saw that the toxicity did not differ significantly from that of untreated. And we saw a non-significant increase in the serum cholesterol levels when mice were treated with HB-beta-CD, possibly allowing us to postulate that the mice or the tumors are getting rid of this excess cholesterol as it is being exported into serum and out of the body in the form of bile. So when cross-comparing the models in this study, what do we learn? We see that HT29 cells are derived from a 44-year-old Caucasian female patient with colorectal adenocarcinoma, and this line was instated in 1964, and it is representative of an early-stage cancer. The data presented in this study was derived from a 29-year-old Caucasian female patient of South African descent with invasive colorectal adenocarcinoma, and this is representative of an invasive as well as a moderately differentiated cancer at stage 2, T3, N0. The MF1 mice model, as previously stated, is just a subcutaneous injection of these HT29 cells, which again are representative of an early stage cancer. So when we cross-compare these models, we see that there are indeed unique responses to cholesterol-lowering therapies, and this can be attributed to factors such as disease etiology, cancer stage, the presence of compensatory mechanisms where we see that cancers that are in late stage do actually display an inherent reliance on cholesterol for its function, and then lastly, the physiological relevance of the model and the study. With this, I'd like to call Prof back to just wrap up the rest of my talk. Thank you for your attention.
Just to wrap up. So organoid models, they are really very useful, as we have briefly demonstrated here, that they can actually help us to understand the biology in the cancer undergoing at that point in time, and also using patient samples. So they have wider usages of organoids have been demonstrated in regenerative medicine, drug screening, biobanking, gene profiling, cancer modeling, and many other processes. So for us, what we are doing with these organoids now, so we have different projects ongoing in the lab. Naaziyah's actual PhD work is focused on co-culturing of T cells with the organoids. And why we are doing this? Because what happens in the cancer cells, that when the T cells are basically there to actually kill the cancer cells in a patient, but the tumor microenvironment is very rich in cholesterol, so it exhausts T cells. So what we are trying to achieve here, or test the hypothesis that we have, that if we give our cholesterol depleters to these cells, then what effect can we actually increase the functioning or the viability of these T cells? So this is Naaziyah's PhD project. We are also in the lab, we are recently starting a project to develop a proof of concept that if we can use these organoid models as a clinical assay for therapeutic screening in colorectal cancer organoids, and can we inform the clinicians about how to use or which drugs they are most suitable for a particular patient type. So basically a precision medicine project. And the third project, which is funded by IDORI, seed funding that we got from IDORI last year, so early this year. So this is like we are modeling HPV-mediated oncogenesis in cervical cancer organoids. So that is the next model we are going to start establishing very soon in our laboratory, so that we can monitor how HPV actually induce cancer in these organoids or in humans. So just to wrap up in the sense is that organoids are an emerging technology, I think emerging in Africa, but everywhere else it has emerged. So there are lots of uses that are coming out, and they are taking the center stage going forward. And they are redefining exactly how the biology and the science is being done. And in this case, you can see that, that this is very close to human trials. They are to restore vision using lab-grown retinas. These are basically organoids, and they are going to test that, inject organoids into human patients so that the retina can regrow. There are actually many uses. This usage has been actually a few years ago in UK. There was a patient, cystic fibrosis patient, and the medical aid was denying treatment to that patient. So what they did, researchers did, they created organoids from the patient, they tested that particular drug and demonstrated that it is going to work on the patient, and medical aid had to pay, and it actually worked on the patient. So these are the real-time uses, and this is another example where FDA for the very first time 2-3 years ago actually bypassed preclinical animal model studies to take a drug tested just based on organoids and organoid on a chip models to directly take it to phase 1 clinical trials. So they are finding their ways into drug discovery and development. So what is the impact on Africa if we have these models? In here, of course, the relevant disease models that are right now that I have demonstrated and showed you, basically the cell lines that we use, they are very, very old. they are not representing the biology of our populations. So we need to find better ways to develop new drugs and to find right treatments for our populations. And organoids can pave that way for us. Secondly, testing of, even if we are not developing drugs, because we don't have that kind of money here in terms of developing new drugs, but what if we are able to test newly marketed drugs on our patients' organoids to test whether they are going to work here or not, before even bringing them to the market. And then, of course, elimination of animal models. This is a huge, huge, huge benefit in order to promote sustainable science. And there's a huge drive to eliminate animal models going forward based on ethical considerations and many other sustainable factors. So organoids are very useful, and they can be easily used for organ-on-a-chip models in microphysiological systems to test drugs. So they are possibly going to replace the way we do things. And how, this is one agenda right now I'm trying to drive at Wits, that we need to set up a central organoid facility at Wits so that we now have the know-how. This is a technology which is not out there in Africa. So that is why we can take a lead on this. We can have this centralized facility so that many laboratories, many people, many students, they can actually use this technology going forward in their research projects, and we can reduce our reliance on animal models. So that would be a huge win-win for us going forward. And of course, that also brings in a lot of benefits for Wits in terms of being pioneer in this field in Africa. So what we are planning, if there would be a lot of interest, from everybody, then we are thinking of doing a dedicated talk on organoids, to talk about organoids and their applications in diverse fields, and specifically linking it with a lot of different types of research projects that are being done at Wits, so that we find some sort of common grounds here and we move forward together. And with that, I would like to, again, this doesn't like me at all. Acknowledgements, lots of acknowledgements are due. I am very thankful to surgeons, Dr. Babington at Wits Donald Gordon Medical Center, and Professor Luvango is sitting here somewhere. Yes, thank you very much. He has been quite instrumental in terms of helping us set up collaborations with Charlotte McKay Hospital, where we have recently started a collaboration. And then, of course, Professor Andrew Beggs in Birmingham, like Naaziyah has been recently trained in that lab for 4 months. And there are many collaborators, different funders, Wits Innovation Center, Wits Commercial Enterprise, a lot of other people. And most importantly, my lab members, my students, very proud of them. They work very hard. And also a special thanks to the patients and, of course, to the animals who have given up their lives for our research that we have shown you here. And thank you very much.
All right, thanks, Mandeep and team. So we can take some questions now. Anyone with a burning question?
I just, out of interest's sake, I wanted to find out about, has the PCSK9 inhibitors been investigated in assessing cancer cell proliferation? And have you done it in your lab, or has it been done internationally, and what were the outcomes?
Yes. That's a very important question. Thank you. Didn't get time to talk about it. So right now the market is very hot in terms of using PCSK9 inhibitors for cholesterol lowering as a cholesterol lowering treatment, especially when statins don't work. But that is what our worry is at this moment, because PCSK9 is it degrades LDLR. That is how it works in the blood. But for cancer cells, you can see that if we inhibit PCSK9, it will actually block degradation of LDLR, which leads to influx of cholesterol in cancer cells and fuel cancer cell growth. So, of course, the studies, when the drug companies, they have brought a PCSK9 into the market, they have not paid attention to what it is going to do in terms of fueling the cancer growth. And there are not many studies that are out there. People have started to touch upon it. Very few. One study just came out And they were not even able to demonstrate that how it will impact the cancer cells. But going forward, we also want to understand and test some PCSK9 inhibitors and see how they are going to impact cancer growth and aggressiveness. But that's something very important to keep in mind when using, PCSK9 inhibitors, specifically in cancer patients. And they might also again lead to increase in cancer invasiveness, such as like we have seen for statins and the molecule that we are testing. Okay.
Thanks very much for that enlightening talk, fascinating. So we see a slightly different model in lung cancer. And in lung cancer, what we see is we see the tumor microenvironment devoid of antigen-presenting cells or of inflammatory cells. And we're not too sure the pathogenesis behind that. We're suspecting it's because the tumor cells produce some sort of signaling or inhibitory signaling substances that actually prevent the influx of inflammatory cells into that particular area. Now, cholesterol molecules are easily identified by the most native of our inflammatory cells, macrophages. I mean, that's how we get atherogenesis and plaque. So I don't know that the same model applies to lung cancer, because I wouldn't anticipate that cholesterol, remnants or even cholesterol itself would actually prevent the influx of inflammatory cells into the tumor microenvironment in lung cancer. So is there something else that we're missing in that pathogenesis, or is it still because of tumor fatigue, because of the cholesterol-rich environment?
It could be, because immune system is very complex, and especially when we are talking of immune cells, inflammatory cells, and in that mix we have T cells working. So there's not a lot known about the concept that we are even testing with T cells. There's just one paper out around this concept. So we really do not understand that how heavily reliant this tumor environment is on cholesterol. We do know it does. It is enriched in cholesterol, but what is the impact of that on different types of cells, different types of diseases, and responsiveness to different types of chemotherapeutic drugs, that are specifically used during that stage. So at this stage, no, I'm not sure, if that can be addressed, yeah.
Would that be one of the reasons why cholesterol would be so high in that particular environment, whereas we're not seeing that same epiphenomenon in other cancer models? It
could be. But again, if I go back to one of my very original slides, I think you were not here at that point in time. So actually, basically, cells produce, or the body produces its own cholesterol. Yeah. And the heavy reliance of cancer cells on cholesterol means they really do not need to take up cholesterol from outside. They inherently synthesize it, so they are not dependent on one process. There are multiple processes in which they are dependent. But that is also known that not all different types of cancers are actually enriched in cholesterol. We know of prostate, we know of esophageal, we know of breast cancer, colorectal, pancreatic. So it could be that lung cancer cells are not really heavily reliant on cholesterol, and they use something very different for their processes. So it's just the basic biology that we need to, of course, understand and see what is happening. Yeah. Thank you.
Is the mitochondrial cholesterol also depleted when you treat the cells?
Actually not. So because mitochondrial cholesterol is not really, it's very little mitochondria, cholesterol is present in mitochondria as compared to rest of the cell. And the studies have shown that, this molecule HP beta CD, it does not really go and impact, mitochondrial cholesterol as such in our case. But we have not tested it.
Lovely work, well done to you and your team. Just a few technical questions. I wanted to ask about, so with the colorectal primary cultures, because of the bacteria present in the gut microbiome, you actually struggle with growing these primary cultures in terms of contamination. What measures did you implement to mitigate that? Mm-hmm. And then the other question is, in terms of the pancreatic cell line that you used was PANC1. Yes. Does that represent the head or the tail of the pancreas? Because in terms of pancreatic cancer, that the disease presents differently in terms of the head and the tail. So what part does it represent, and would you anticipate a different result if you use a different pancreatic cell line?
Okay. So definitely, for the very first question, I'm going to ask Naaziyah to respond to that, because she's definitely an expert, but I do know we do use antibiotics in there, but she can actually explain the process to you.
Thank you for your question. So we use DPBS supplemented with Aronson that has antibacterial and it also has antifungal activity. And we've been very successful in mitigating contam that way. In our tissue storage solution, when we collect the specimen from the hospital, that's supplemented with Aronson. When we bring the tissue specimen back into the lab, we wash it thoroughly with PBS supplemented with Aronson. And our initial culture media also has Aronson in it. And following that technique has proven very successful to mitigate the growth of any bacterial or fungal contact.
And secondly, for PANC1 cell line, definitely, if we use different cell line, these different cell lines represent different stages sometimes, aggressiveness of the cancer and different type of disease as well. Of course, we are going to get bit of different results when you use different type of cell lines because they have different mutations in them as well. And in case of pancreatic cancer, like 85% of cancers, they are present in the head. The ones that are present in the tail, they are very difficult to detect, difficult to operate because of their location, and they are not really, that prevalent. So PANC1 is from the head of the pancreas. So that is the most we normally work for the treatment for the most common types that are existing out there. But you are right, they are going to give us very different results, and you can see here even different types of animal models, you get different results. So there are hundreds and hundreds of limitations that are linked to each cell line, each animal model, and even with organoid model, they would be. So from where we are taking the tissue for growing organoids? Are we getting enough stem cells in there? What is the disease stage of the cancer? If we are choosing to grow organoids from, for example, a very aggressive or a late stage disease, then we are going to see very different results as compared to when we use the early stage disease. So yeah, so there's a lot we need to wrap our heads around. But thank you for your questions.
Thanks so much, Prof. I have two questions. Firstly, I'm interested in your hydroxypropyl beta-cyclodextrin molecule. Are there any studies on how big the cavity is? Because there were very impressive results when you used it co-delivery with other chemotherapeutic drugs. So could there be any interactions with the drugs,
Yes.
possibly reducing the side effects? And like you said, the animals could survive for much longer. And secondly, with regard to the organoid model, the move towards the removal of in vivo studies. Have you thought about pharmacokinetic studies, to supplement the organoid studies to check how the treatment would release and be metabolized in the treatment?
So there are different types of cyclodextrins, like alpha, beta, and gamma. So beta is like depending on how many glucose units are in there, their cavity size is determined. So I cannot remember the actual size, but you are right, they bind to different types of drugs. There are loads and loads and loads of studies, especially in your field in drug delivery. Cyclodextrins are sometimes the molecules of choice for delivering drugs to various places. So yes, they do bind to different drugs. We know they bind with the paclitaxel, they bind with 5-fluorouracil. Load of in vitro work is there, and that is how they increase the solubility of these drugs and accessibility to these drugs, and that is why the side effects are also reduced. So that is one thing. For organoids, yes, that is the next step. Like once we are there, then to do some sort of pharmacokinetic studies and see exactly what is happening to these drugs. And recently, I think last week, we just visited Department of Physiology as well. We are planning to use that MS imaging as well, to see that how our drugs are actually, especially our molecule, is penetrating into the organoids, and if we can build from there as well. So yeah, so those things are in the thinking, not in the pipeline.
I was wondering how we can involve, how can we involve or look at the effects of the stromal cells, the stromal layer, the signals that arise from those cells on the tumors, on the organoids as well. Because, for example, with pancreas, the stromal layer plays quite an important role in signaling the tumor. So growth signals arise from the stromal layers.
So what we have observed in our cultures, and it's a very hard learned lesson, if I say so. When we get tissues to grow our organoids, and we have seen that when we get more stromal tissue, like sample in there, organoids do not grow at all from that. So that is a limitation in terms of, growing organoids if we do not have a clean tissue. And I think this is something we need to really look forward to going forward, that how, to be very honest, I don't know how we can, merge these two and study. But yeah, that would be very interesting to study going forward. But again, the opportunities are amazing here because we are just starting up in this field as well. So we really need to learn a lot going forward around that.
Well, Mandeep, just remains for me to thank you and your team for telling us about your bedside or theatre to bench approach, and hopefully back again eventually to the patient at the end of the day. I think it's a very interesting topic and very compelling and worthwhile endeavour. And we hope to hear, as you say, we're going to hear from you again in the not too distant future. So thanks very much.